Hi everyone, this is 10th video in our Advanced Pharmaceutical Dissolution Science and Application series. In this video, we will discuss understanding drug release mechanism, diffusion, erosion and osmosis. Do you know how a drug is actually released from a dosage form? Understanding drug release mechanism is essential for dissolution method development, formulation design, IV/IVC and regulatory justification. In this video, we will demystify how drugs are released through diffusion, erosion, and osmosis. This knowledge will help you to design better methods and make stronger scientific decisions. Understanding drug release kinetics is the foundation of dissolution method development. It helps build robust QC methods, identify formulation risks, and optimize product design. Drug release mechanisms allow us to predict real-world product performance and guide critical choices, media selection, apparatus type, and sampling time points. This understanding is essential for immediate, sustained, and controlled release formulations, quality by design, IB/IBC development, and regulatory submissions. Simply put, if you understand how the drug is released, you understand how it will behave in the body. Most solid oral dosage forms release drug through one or a combination of three mechanisms. Diffusion. The drug diffuses out the matrix. Erosion. The matrix gradually dissolves or breaks down. Osmosis. Drug release is driven by osmotic pressure through a semi-permeable membrane. Many formulations use hybrid mechanisms. For example, gel-forming matrix that rely on both diffusion and erosion. Understanding these mechanisms helps explain why formulations behave differently in dissolution testing. Let's briefly talk about diffusion controlled drug release. No equations needed. In these systems, polymer swelling forms a gel barrier and the drug slowly diffuses through this layer from higher concentration to lower concentration. The release rate depends on polymer viscosity, drug solubility, particle size, and degree of hydration. This process follows Fickian diffusion principle and is commonly seen in hydrophilic matrix tablet, coated pellets, and gel-based system. Understanding diffusion helps explain why release is gradual and predictable in these formulations. In diffusion-controlled systems, the release curve is not linear. As time passes, diffusion path becomes longer, so the release rate gradually slows down. In HPMC-based hydrophilic matrix tablet, the hydrated polymer forms a gel layer that regulates drug release. This behavior typically follows square root of kinetics as described by Hughes model. That's why diffusion system shows fast release early followed by a slower controlled release over time. In erosion controlled release, the matrix itself gradually breaks down, releasing the drug. Unlike diffusion, drug release here depends on polymer or matrix degradation, not movement through the gel layer, making it especially useful for poorly soluble APIs. There are different erosion mechanisms. Chemical erosion driven by hydrolysis, enzymatic erosion driven by biological enzymes, dissolution-based erosion where the matrix simply dissolves. Erosion can occur as surface erosion seen with material like polyanhydrides, bulk erosion common in PLA-PLGA systems. This approach is widely used in controlled release tablet and system where diffusion alone is not sufficient. In erosion-controlled systems, the dissolution medium plays a major role. Media pH can accelerate or slow polymer erosion, directly affecting the drug release rate. Common erodible matrices include carbomers, HEC, and xanthan gum. When polymer breakdown is consistent, erosion system can deliver a linear or near constant release profile. However, release behavior is highly dependent on polymer composition and solubility and is sensitive to pH and hydrodynamics. That's why erosion-based formulations require careful media and method selection. In osmotic drug delivery systems, release is driven by osmotic pressure, not by erosion or diffusion. Osmogens like sodium chloride, potassium chloride, or sugars attract water into the tablet through a semi-permeable membrane. As water enters, osmotic pressure builds and the drug is pushed out through a laser-drilled orifice. This design delivers a highly controlled, nearly zero order release and is largely independent of GI pH, mobility, or food effects. Well-known examples include glucotrol XL and Procardia XL, both based on Oros technology. That's why osmotic pressure are among the most predictable controlled release platforms. Osmotic drug delivery systems offer major advantages of IBIBC development and regulatory confidence. They can achieve near zero order release with highly reproducible performance that is largely unaffected by pH, agitation, and GI variability. This predictability comes at a cost. Manufacturing requires specialized components such as a cellulose acetate semi-permeable membrane and precise laser drilling of the release orifice. So, while osmotic systems are robust and reliable, they also involve greater manufacturing complexity. That's the trade-off behind one of the most predictable controlled release technologies. Designing an effective dissolution method starts with understanding the dominant and secondary drug release mechanism. In practice, most real products use mixed mechanism, not just one. That's why hybrid systems may require multi-stage media or specialized apparatus to capture true release behavior. For example, an HPMC matrix releases drug by diffusion plus erosion. An osmotic core with a polymer coating combines osmosis and diffusion. Recognizing these combinations help you to select the right medium, apparatus, and sampling strategy. They guide critical choices such as medium selection, apparatus type, rotation speed, and sampling strategy. For example, diffusion control systems are sensitive to agitation speed, erosion control systems are sensitive to pH and media composition, osmotic systems show minimal sensitivity to hydrodynamics. Because of this, release mechanism determines which medium to use, which apparatus to select, where to place sampling points, and how discriminatory the method will be. Most importantly, understanding the mechanism of the foundation of IBIBC, regulatory justification, and risk assessment. In real life, across QC, R&D, and regulatory filings, understanding drug release mechanism makes all the difference. To recap, diffusion. Drug moves through a hydrated gel layer. Erosion. Matrix breakdown controls release. Osmosis. Pressure driven near zero order release. When you know how a drug is released, you can design better formulations and more scientific, robust, and regulatory friendly dissolution methods. Knowing the mechanism means better decision and better product. Thank you for watching. If you found this explanation helpful, please like, share and subscribe. In the next video, we will cover media degassing techniques, best practices and common failures. And stay tuned because in this 62 video series, we will deep dive into dissolution method development, validation, troubleshooting, advanced approaches and real industry case studies. If you have any question, please post them in comment section. See you in next video. Thanks.